So I'm very happy to introduce our seminar speaker this week, Stewart Merrill. She has probably a somewhat different background than most of our seminar speakers that we've had this year, in that she has 2 bachelor's degrees, and her first one is in dance. And then she spent several years as a professional dancer before then going back to get a second bachelor's degree from Santa Barbara in biology. So I think that will come across in her presentation. She gives amazing talks, has great presence. And so after she got her bachelor's in biology, she was a lab tech for Armin Curris and Kevin Lafferty. And so she was by then fully into parasite land. And she then moved to the University of Illinois, where she was a PhD student with Carla Caceres, who's a long-term collaborator of mine. And she got her PhD in 2019 and now has this superstar postdoc fellowship, so Life Sciences Research Foundation postdoc. And her postdoc is supported by the Simons Foundation. And she's doing that at the University of Colorado, where she's working in Pete Johnson's lab. And so Tara does really impressive work looking at immunity and trying to understand the causes and consequences of that. And I will hand it off to her so she can tell us about that.
Well, thank you, Meg, so much for that introduction, and thanks everybody for coming today. It's been such a pleasure meeting faculty and postdocs and graduate students here at University of Michigan, and just learning more about this thriving research community and academic environment. So again, my name is Stewart Merrill. I'm a postdoctoral fellow at the University of Colorado Boulder, and I'm a Simons Foundation fellow with the Life Sciences Research Foundation. And today I'm going to be talking about variation in host immunity and what that can tell us about disease dynamics in natural systems, and in particular in freshwater ecosystems throughout North America. So before I begin, there's a huge group of people I'd like to acknowledge, including my colleagues, my research assistants, as well as my funding sources. And thanks especially to 3 of my mentors, Carla Caceres, Peter Johnson, and Kevin Lafferty, as well as to Meg for inviting me out to speak today. So my journey into ecology began over a decade ago. And from the very beginning, I was interested in exploring aquatic ecosystems. So I got my start working at Carpinteria Salt Marsh in California. I then studied plankton in Lake Gatun surrounding Barrow, Colorado Island. For my dissertation work, I worked on 6 temperate lakes in central Indiana, and now for my postdoctoral work, I'm studying the ecology of several freshwater ponds in California. And what I really love about aquatic ecosystems, and what largely attracted me to their study in the first place, is that I really believe that aquatic systems exemplify sort of the mystery of ecology. And what I mean by that is that when we go out and we visit a body of fresh water, we're often met with this very glassy surface, below which we know many different hidden interactions are occurring just out of our view. And our various limnological methods provide glimpses into what might be going on below the surface. We can then string together those pieces of information to generate ideas, to test theory, and to try to uncover a cohesive understanding of how these systems work. Now, adding up all of these glimpses through time, we've learned that freshwater ecosystems include tremendous diversity, complex but elegant food webs, as well as this strong connection between the biological and the physical environment. But following the generation of this sort of picture, I'm still interested in going even deeper into the hidden interactions within this system. And for me, that means looking inside aquatic organisms and uncovering their hidden parasites. So parasites and the diseases that they cause can be as diverse as the free-living hosts that they're found within. And really, the central motivating question of my developing research program is, what are the causes and consequences of parasites in freshwater ecosystems? So how do parasites fit into our understanding of community structure and ecosystem function? And importantly, how do aspects of the aquatic system itself regulate the transmission of parasites and infectious diseases? These are important questions to ask because many species, both aquatic and terrestrial, require freshwater for their survival. And freshwater can therefore serve as a nexus of parasite transmission. Now, ultimately, many of these diseases can have devastating consequences for wildlife. So for instance, VHS virus has decimated fish populations across North America. Chytridiomycosis has led to global declines in amphibian populations, and crayfish plague has led to dwindling stocks of crayfish throughout Europe. So parasites can be problematic. But parasites are also thought to be important players in ecosystems and in community-level dynamics. So if we were to go out to a large body of fresh water, for instance, Lake Michigan, we might find yellow perch there. And an important terrestrial predator of yellow perch is the double-crested cormorant. Now, yellow perch, as well as other fishes, can be parasitized by a parasite called clinostomum, or yellow grub, and this parasite causes these large white blobs on the fish when it insists on fish tissue. Now, those white spots make the fish more conspicuous to bird predators, such that this parasite increases the likelihood that an infected fish will be consumed by a bird. So if and when this parasite is present within a system, it can strengthen this trophic link, increasing the flow of energy from fish to birds. In Lake Tokvatn in Norway, the effects of parasites on ecosystems and on food webs have been resolved at a much larger scale. So here, researchers at the University of Tromsø assembled a pelagic food web that contained all of the free-living species within a system. And then they compared that food web to one that also contained parasites, shown here in red. And adding parasites did a number of things to the food web. So it increased the number of trophic levels, it more than doubled the number of trophic links, and ultimately increased food web connectance. So parasites can be problematic when they wipe out host populations, but they're also thought to be important players in ecosystems. And it's really these two factors together that have led to my central question of what are the causes and consequences of parasites in freshwater systems. And today I'm going to focus on that first half of the question. So what are the causes of disease? And to ask that question, I'm going to use the zooplankton Daphnia. So Daphnia are keystone players in lake food webs. They're the dominant algal grazers, and they also represent an important resource subsidy for young of the year fish. But Daphnia aren't just eaten by fish, they're also eaten by parasites. Now, many of these parasites are what we call castrators and killers. So castrators totally eliminate the Daphnia's reproductive capacity during the course of infection, and killers have to kill the Daphnia host in order to be transmitted. So these killers essentially fill up the Daphnia's body with tons of infectious propagules, and then that Daphnia essentially explodes, releasing those propagules back to the water column. Now, given the strong effects of many of these parasites on individuals, some of these parasites can also have suppressive or regulatory effects on Daphnia populations. And given Daphnia's keystone role in lake food webs, I think it's therefore important to ask who these parasites are, how strong their effects are, and also under what conditions they spread. So I study a killer of Daphnia, a fungal parasite called Mechnicauia bicuspidata. And if we look at the prevalence of this parasite across separate lake ecosystems, it's very common to find that this disease varies both in space and through time. And the major question that I'm going to focus on today is what's driving this variation. So what conditions lead to the spread or the lack of spread of this disease in Daphnia? So first principles tell us that disease is regulated by a simple set of factors. In particular, disease is thought to result from interactions between pathogen exposure and susceptible hosts within an environment. Now, although this theory is simple to describe, it's actually been pretty challenging to empirically quantify some of these processes in natural systems. And in studies of wildlife disease, susceptibility is a notorious black box. So many studies of wildlife disease go out into the environment and measure pathogen exposure to see how exposure mediates transmission, but susceptibility is often unmeasured and in many cases just entirely overlooked. Now, this focus on pathogen exposure is not unwarranted, as exposure is totally critical for the spread of parasites and pathogens that cause disease. And this is very clearly evidenced in what's known as the dose-response curve. So essentially, as you increase the number of parasites or pathogens a host is exposed to, that host's probability of infection increases. So a sometimes relevant example is if you go out and meet one friend for coffee and that friend has the flu, that's a lot less risky than hanging out in an auditorium full of flu-infected people. So given the nature of this curve, a common assumption has been that a population facing a certain level of exposure might have a predictable prevalence of disease. But that assumption doesn't really hold up if susceptibility varies. And as it turns out, in the Daphnia system I study, we see tremendous intraspecific variation in susceptibility. So shown here in red is the average dose-response curve for Daphnia, with all of the different points contributing to that average coming from different populations and points in time. Now, some might argue that to understand disease, we really only need to know the shape of that red curve. And the question that's really motivated my research over the last few years is whether that's a safe assumption to make. So as we try to both predict and understand disease, can we safely ignore all of this variation in host susceptibility? I'm going to address that question today in 3 parts. So in the first part of my talk, I'll unpack susceptibility and ask, what is susceptibility, and how do we best measure it? In the second part of my talk, I'll explore how susceptibility is mediated by environmental conditions. I'll focus on the flexibility of host immunological traits and ask, how is host immunity shaped by broader host ecology? Finally, I'll take both of these pieces of information into a set of natural lake ecosystems and ask, how do exposure and susceptibility interact to either promote or inhibit the spread of infection? And to do this work, I'm going to build up from ground zero. So I'm going to first focus on the natural history of this system, as well as some mathematical models. I'll then move to data from laboratory experiments and field observations. And then finally, I'm going to tie it all together with a 6-month field study. So when I started research on Daphnia, we knew very little about the basis of their susceptibility to things like castrators and killers. And I really wanted to know, first, can Daphnia recover from infection? And if so, how do they do it? So in this first section of my talk, I'll describe how I coupled natural history with a simple modeling approach to try and quantify susceptibility, as well as the host traits that underlie susceptibility. And I'd like to acknowledge my co-authors, Carla Cáceres and Zoe Rapti, as well as 4 amazing undergraduates who contributed to data collection for this study. So like I mentioned, I study a killer of Daphnia, a fungal parasite called Mechnicauia bicuspidata. And Daphnia become infected when they inadvertently consume fungal spores while they're filter feeding. Those fungal spores then develop and reproduce inside of the Daphnia, and then that Daphnia dies and releases those spores back to the water column, where they can go on to infect more susceptible hosts. Now, this host-parasite interaction, it occurs in a natural context. It occurs in temperate lakes across North America, and we have very valuable information regarding the role of different ecological processes, like predators, resources, and even like physical structure to the spread of this disease. But when I entered this system, the within-host traits that comprise a Daphnia's susceptibility were pretty much a black box. So we knew that if you fed some Daphnia some fungal spores, 10 days later, some of them would be infected and eventually die, and others wouldn't. But we really knew very little about the host traits that govern this parasite's establishment, growth, and reproduction. But the real power of the Daphnia system is that Daphnia are transparent, which makes it relatively easy to open this black box. So by just looking inside the Daphnia, I could watch this parasite's life cycle unfold in front of my eyes and actually observe all of the different ways that the host tries to fight back against this infection. So now I'm going to try and uncover this hidden interaction for you and show you what happens after Daphnia consumes these infectious fungal spores. So first, the spores, they're shaped like little needles. And when the Daphnia consumes the spores, those needles penetrate the Daphnia's gut and move into its body cavity. So here you can see one of those spores that has crossed the gut epithelium, has moved into the body cavity, and has host immune cells, or what we call hemocytes, that are aggregating on the spore and trying to kill it. Those spores then emit fungal hyphae, and these hyphae grow out laterally, like finger-like extensions. So they adhere to inner surfaces within the Daphnia's body, and then they start to gradually grow as they assimilate host resources. Those hyphae then develop into sporocysts, or small sacs, and we typically see these sporocysts residing in the Daphnia musculature. So here up above is this nice muscle tract, healthy striated tissue of Daphnia, and this more necrotic tissue below is a muscle tract that's been overtaken by dozens of these Mechnicowia sporocysts. Those sporocysts then rupture and release conidia, and these conidia are the typical yeast form of the parasite. So they reproduce by budding, they disperse through the host, they continue budding, and this is how we see the fungus build up really high-intensity infections within its host. Then finally, those conidia elongate to form asci, or large sacs, and each ascus houses one infective spore. So here you can see a couple of these spores under high magnification, but if we were to zoom out, we would see that the Daphnia is totally full of these fungal spores. All of that brown material represents the fungal parasite, and an infected individual can yield in the tens of thousands of these fungal spores. So breaking down this binary infected yes or infected no response and trying to inform it with natural history has revealed these 5 distinct within host stages that provide a really nice framework for thinking about why Daphnia might vary in their susceptibility. So for instance, Daphnia can limit that initial spore puncture by increasing the resilience of their guts. And that initial prevention of infection is what I'm going to refer to today as resistance. Now, if a Daphnia can't prevent infection, it can kill any of the subsequent infection stages with an activated cellular immune response. And this killing of an infection is what I'll refer to today as clearance. So a few years ago, I took a population of Daphnia, I exposed them to this fungal parasite, and then I tracked their infection stages through time. And I was able to look at each Daphnia at 3 different time points over the course of infection. So I looked at them all at time 0 when I fed them spores. I performed their second examination at some point in time between days 2 and 8 post-exposure. And then I examined them all again at day 10 post-exposure. On the y-axis, we have all of the infection stages that I just showed you, and you don't need to worry about keeping track of them, as long as you remember that moving up the figure means moving through the infection process. So at time point 0, I fed my Daphnia spores, they were exposed, and then they moved through these various infection stages, ultimately converging on that final ascus stage that will kill them. But I've left out a little bit of data here, and if we include the full complement of data from this study, we can see that some Daphnia individuals moved from exposed to uninfected, they resisted their infections, whereas others moved from various infection states back to the uninfected class. They cleared their infections. So now I can see that recovery is occurring, but how do I quantify these 2 different forms of host trait? Well, with this sort of data, some simple division would allow me to calculate the probabilities with which these phenomena occur, but I can't really do that in systems beyond Daphnia because most organisms aren't transparent like Daphnia. So if I wanted to do this in a fish or a bird, I would have to dissect the host to stage its infection, and dissecting it means killing it, which means that we can't track the progression of infections through time. So one solution to this is to use cohort studies. And here I'm going to put up a separate population of Daphnia for you. But rather than track individuals through time, I can track how cohorts move through time. So on the x-axis, we have the days post-exposure to the fungal parasite. And on the y-axis is the proportion of each day's cohort that's occupying a given state. So I take a group of Daphnia, I stage their infections, I throw them away, and then I start the next day with a new group of Daphnia. Now, Daphnia can occupy any of the infection states that are shown above, and I've also included an exposed state where Daphnia have consumed spores, an attacked state where those fungal spores are attacking the gut barrier, as well as an uninfected state and a dead state to account for mortality. So at time point 0, I feed all of the Daphnia fungal spores, and 100% of them are in the exposed state. 1 day later, some of them are being attacked by spores. Some have spores that have moved successfully into the body cavity. Others have progressed to hyphae infections, and some of them have died. And we can continue tracking the progression of cohorts through these stages through time, and importantly, still capture that signature of recovery as we look towards how the proportion of the population that's uninfected increases through time. So now I have an approach, a longitudinal approach that's suitable for systems beyond just Daphnia. But looking up here, I've still been working with this rather complex 9-state system when I'm really only trying to quantify 2 forms of host defense. So I've taken a step back, boiled down some of this complexity, and condensed the system into a more translatable or general 4-state system. So here we have 4 key epidemiological states. We have infected in red, uninfected in blue, exposed in yellow, and dead in gray. And to analyze these longitudinal data, I've developed a Markov model. So a Markov model is a stochastic model used to measure transition probabilities among separate states within what we call a fixed state space. So here we have those same key states: exposed, infected, uninfected, and dead. And with this model, we can actually measure the probabilities of transitions among them, like the probability of infection, the probability of mortality, the probability of remaining in a state, and importantly for my research, the probability of recovery. So animals can move from exposed to uninfected via resistance, and in this system, I think that's driven by a robust gut epithelium. And animals can also move from infected to uninfected, via clearance, which I think is driven by an activated cellular immune response. So Daphnia are clonal organisms, and we maintain different clones or genotypes in the lab. And these genotypes vary in their susceptibility. So if we take several of these genotypes and we expose them to the exact same dose of fungal spores, some of them are going to have low resulting prevalence, and others are going to have high infection prevalence. And I really want to know why. So do immunological defenses underlie these differences, and can I explain all of this variation using only resistance and clearance? Well, now we can take this Markov model, and we can apply the model to different genotypes, apply it to their longitudinal data, and ask, well, does this low susceptibility clone just have higher levels of resistance and clearance than a high susceptibility clone? So I've done that for 6 separate genotypes of Daphnia, and plotted on the x-axis are their Markov model estimated probabilities of resistance, and on the y-axis are their empirical prevalences, their susceptibilities to infection. And if we look at resistance, there's not the strongest relationship between resistance and prevalence, suggesting that resistance doesn't explain a ton of the variation in their susceptibility. But if we turn our attention to clearance, we can see that there's a strong relationship between each clone's probability of clearance and its susceptibility to infection. So this clone here that is highly susceptible to infection, we can see in its longitudinal data that very few animals ever enter the uninfected class, and the Markov model tells us that it has a low, near zero probability of clearing infection. Whereas this clone over here, this clone has very low susceptibility to infection. And there's a signature of clearance in its longitudinal data, and the Markov model tells us that it has a 60% probability of clearing an infection. So in asking the question of why Daphnia vary in their susceptibility, I've taken this complex host-parasite interaction, broken it down into the key steps of infection, and then leveraged those steps to build a model that can broadly quantify 2 forms of host defense. And doing this taught me 2 pretty important things. First, that Daphnia can recover from infection with their killer parasites. And second, that Daphnia vary broadly in their abilities to resist and clear infections. Now, all of the data that I just showed you suggests that there might be a genetic component to this variation, but I next really wanted to know whether this variation extended beyond genotype. So are resistance and clearance flexible? And if so, how are they mediated by environmental conditions? In this next section of my talk, I'm going to move away from the modeling framework and instead focus on data from laboratory experiments and field observations. And rather than focus on those Markov model probabilities, I'll be using a trait-based approach to see how susceptibility is mediated by environmental conditions. And my co-authors for this work are Carla Cáceres, Spencer Hall, and Ilona Manal. So we sometimes have to remind ourselves that with the exception of some cockroaches, labs are just not natural habitat. But rather, our study species are members of complex ecosystems, and their interactions with biotic and abiotic factors have the potential to influence host-parasite interactions in potentially unique ways. So let's consider resources for a second. So resources are required for an organism's physiological functioning, and a common prediction in ecological immunology is that as you increase a host's access to resources, that host will be able to mount more robust immune responses. But the outcome of host-parasite interactions along a resource gradient might not be so straightforward. And that's because parasites, by definition, feed off of host energy, such that as you increase host access to resources, that host might also better fuel parasite growth. Now, I mentioned resources because they're not an arbitrary factor in plankton. So Daphnia and other species of plankton spend the majority of their time filter feeding, and the phytoplankton that they consume can undergo these large and regular changes throughout the year. Now, these gray shaded areas represent phytoplankton biomass over the annual cycle in two types of lake ecosystem. And in addition to major changes in biomass, we also see major differences in resource quality, both within lakes and among lakes. Now, if resources mediate host immunity, and if resources mediate within a host parasite growth, then any changes to resources within an ecosystem might be powerful predictors of whether or not disease can spread within a system. So I conducted an experiment looking at how Daphnia hosts and this fungal parasite respond to low, medium, and high levels of resources. And here's where we get into that trait-based approach. And I really want to focus your attention on those needle-shaped spores entering the Daphnia's gut, because that's where much of the action happens in this system. So earlier I quantified the probability of resistance for you, and one way that we can empirically measure resistance is by just counting all of the spores that attack the Daphnia's gut, and then keeping track of the subset that successfully cross through the gut and enter the body cavity. So when taken together, these two values give us an idea of the gut's penetrability, which is a key resistance trait. So Daphnia that have impenetrable gut barriers are far less likely to become infected than those with penetrable gut barriers. Now, once those spores enter the body cavity, they're met with an activated cellular immune response. And if we count all of the immune cells that respond to a given level of infection, we arrive at a clearance trait, where Daphnia that mount higher cellular responses are also less likely to succumb to infection. So what happens if we vary resources and look at these two forms of defense? Well, to get at gut penetrability, I regress all of the infecting spores against all of the attacking spores. Here, a 1-to-1 line indicates no resistance, but as this slope tends to 0, resistance of the gut increases. And it turns out that the gut's penetrability doesn't really vary among low, medium, and high resource conditions. And this, paired with some of my other data, suggests to me that the gut and its structural integrity may be somewhat genetically predetermined and may be somewhat resilient to environmental conditions. But somebody should definitely ask about this at the end of the talk, because Meg and I have some very interesting collaborations going on with this. Now, to get the hemocyte response, we have to keep track of the level of infection. So I look at all of the infecting spores, and then I regress on the total number of recruited hemocytes. And it turns out that hemocyte production does vary by resource treatment, with hemocyte production being significantly reduced within a low resource environment. And this really supports the idea of an energy-dependent immune response in Daphnia. Now, I would like to rerun this at some point along a much finer gradient of resources because it looks like we might have hit a threshold where once Daphnia move into this medium resource condition, they're already operating at their highest levels of immune response. All right, so let's see what all of this means for the parasites. Well, at first glance, resources don't appear to matter. So infection prevalence does not vary by resource treatment. But parasite prevalence doesn't tell the full story. So for the Daphnia host, it does, because whether or not a Daphnia is infected is literally a life or death situation. But the parasite's fitness is not just whether or not it can infect a host, but also how many fungal spores are produced from that infection. And if we turn our attention to spore production, there's a clear signal that Daphnia with access to greater numbers of resources tend to produce far fewer fungal spores. And my interpretation of this is that the hemocyte response differences that I observed might not have been different enough to create differences in infection outcomes, but they were different enough to alter the course of parasite growth. Now, this is just a back-of-the-envelope calculation, but to give you an idea of what this might mean for transmission, I've multiplied the average spore yield in each treatment by the total number of infections produced in each treatment. And here we can see that cumulative spore yield decreases with increasing resources, suggesting that the resource environment, through its interactions with host immunity, can modulate parasite growth and hence future risk to other hosts. So in summary, changing up resources does not change a resistance trait, but it does change a clearance trait, and there are consequences of this for parasite growth and potentially parasite transmission. Now, all of the data that I just showed you were collected in a superficial laboratory experiment, and I think a really important consideration moving forward is whether the range of resource variation I manipulated is similar to what Daphnia might experience in the wild. And one way that we can start to get at this is by looking at their egg ratios or their clutch sizes, which are a proxy for resources. So here in my superficial laboratory experiment, we can see that clutch sizes varied from about 2.5 in the low resource treatment to about 5 in the high resource treatment. But if we look at clutch sizes from wild-caught Daphnia, those that I collected from lakes, we can see that their clutch sizes are on average much lower, and that wild Daphnia have a much greater range of variation in their resource availability. And for me, this was great motivation to go out into these natural systems and say, okay, what do these two immune defenses, the hemocyte response and gut penetrability, look like out in the wild? So I visited a set of lakes in central Indiana, and I collected plankton. I brought them back to the lab, I exposed them to the parasite, and then I measured these two separate forms of immune defense. And here, each color represents one of the 6 lakes that I sampled, and we can see that for both gut resistance and the hemocyte response, Daphnia exhibit high levels of spatial and temporal variation in these 2 key immune defenses. And I don't really have time to talk about the roots of this variation today, but I do have a number of collaborations in the works looking at how things like lake resources, co-infection with other parasites and pathogens, lake temperature, and even population genetic structure are all contributing to this high degree of variation. So I've taken 2 of the traits that underlie susceptibility in Daphnia, and I've looked at them under both experimental and natural conditions and ultimately found that Daphnia susceptibility is highly dynamic. And so how susceptibility changes, both in space and through time, could influence whether or not disease can spread in a system. Daphnia in natural lake systems can experience annual epidemics with Mechnicowia, and these epidemics serve as nice, tractable units of replication for disease spread. So here on the x-axis is time, and on the y-axis is the prevalence of Mechnicowia within a population. And each colored line represents one of the 6 lakes that I sampled in 2017. And while these lakes very much vary in both the size and the duration of their epidemics, they share this common pattern where for the majority of lakes, epidemics tend to peak right here in early fall, right around the month of October. And things start to get interesting when we look at what's going on with Daphnia immune responses at that same time. So, for instance, if we look at the average hemocyte responses of Daphnia across lake populations, we can see that this epidemic peak tends to coincide with the time when hemocyte production is at its very lowest. And I found this to be great motivation for asking whether all of this individual variation in immunological function could scale up to affect broad patterns of disease within this system. So there are several ways that we can describe epidemics. And what I'm really going to talk about today is epidemic emergence, or the invasion and beginning of an epidemic. And to do this, I'm going to bring together all of my immunological data, and then I'll bring together all of my epidemiological data, and I'll identify when epidemics started in these lakes, and then try to determine the set of conditions that led to their emergence. So we've made it to the third portion of my talk in which I'm essentially asking, can I predict epidemics based on first principles? So can I predict epidemics by only knowing pathogen exposure and host susceptibility within the environment? My co-authors for this work are Carla Cáceres and Spencer Hall. And then thanks also to 4 amazing undergraduates who are going to be future co-authors on a study resulting from this data set. So kind of the study design for this project was I visited 6 lakes in Indiana between June and December 2017. And I visited each lake every 2 weeks, and I brought Daphnia back to the lab, and then I used observation and exposure to try and get at their levels of observation and experiment to try and get at their levels of exposure and susceptibility. So to measure exposure, I measured the prevalence of early infections in the field, and I also, for all of the Daphnia, counted the number of fungal spores attacking the guts of wild-caught hosts. And then to get at susceptibility, I measured the prevalence of late transmitting Mechnicowia infections, and also used infection assays in the lab to generate dose-response curves, and those will become clear soon here. And ultimately, I processed just under 7,000 Daphnia from the pre to the peak to the post-epidemic period. Alrighty, so what can early infections tell us about exposure and susceptibility? Well, I showed all of you the life cycle of Mechnicowia earlier, and I mentioned that on average, it takes about 10 days for a Daphnia to have a fully developed and detectable infection. Because this final stage of infection, the ascus stage, is easy to diagnose, this is the stage of infection that's been traditionally used to measure prevalence and to describe epidemics. But now, all of us know that there are 4 earlier stages of parasite development. And if we layer in those parasite stages, we get a much different picture of this interaction, one in which a sizable portion of the population harbors these cryptic and invisible infections. And we can use these early stage versus late stage infections to try and start disentangling exposure from susceptibility. So for instance, here I've plotted my 6 study lakes for you, as well as the prevalence of infection through time. And all of that red shaded area represents those late detectable infections that are a sign of the parasite's success. So what happens if we add in the early infections? Well, we can see that our first lake, Beaver Dam, has low levels of early infections that never result in late infections and never result in an epidemic. Our next lake, Benefiel, has a huge prevalence of early infections that do seem to correspond well with the prevalence of late infections. And we can keep looking at this lake by lake, and the first thing that's obvious is that when we only look at late infections, we are dramatically underestimating the abundance of the parasite in this system. And this is actually a finding that I've also observed in tropical plankton. But the second interesting takeaway is that early infections can appear long before epidemics actually get going. And we can sometimes witness instances where epidemics failed to emerge, sometimes multiple times. These data qualitatively tell me that pathogen exposure alone cannot predict epidemics and the spread of disease. And we can further unpack this by looking at the relationship between spore exposure, or again, the number of fungal spores contacting the guts of wild-caught hosts, and infection prevalence during the pre-epidemic period, so before epidemics get going. So here, there's a very tight relationship between the level of exposure in a population and the prevalence of early infections, suggesting that if fungal spores are in the environment, they enter their hosts and they begin developing inside of them. But there's no such relationship between those same levels of exposure and the prevalence of late infections, which really suggests that these early infections are not developing into late infections. So where are they all going? I showed you evidence earlier that Daphnia can recover from their infections in the lab, and it could be that before epidemics emerge, Daphnia are just pretty great at recovering. And so by measuring their natural rates of recovery, we can explicitly address how susceptibility is contributing to this pattern. And I'm going to use this lake here, Midland Lake, as an example to build up my framework for you. So in Midland Lake, spore exposure is somewhat gradual, somewhat constant through time, and gradually increases as we move from summer into fall. And I also measure recovery in Daphnia. Now, this recovery value is essentially how many fungal spores a Daphnia can withstand while still retaining a 50% probability of recovery. So it's what we call the half saturation constant from their dose response curves. And here we can see that recovery is declining throughout the season. So Daphnia are getting more and more susceptible as we transition from summer into the fall. And things start to get really exciting when we overlay the epidemic data, because it turns out the epidemic is constrained to this region in time where the natural levels of exposure are higher than the individual's capacity for recovery. So I observed this same pattern in each of my 6 lake ecosystems, and I've implemented the following analytical approach to try and analyze these data. So in this approach, we start with a gradient of spore exposure, which can vary from low to high, and then we layer in recovery, which can vary from high to low. Now, the straightforward prediction is that when the average level of recovery in a population is higher than the level of exposure, an epidemic cannot begin. But as exposure matches or exceeds the level of recovery, then an epidemic can succeed. So I just take the difference between these two values, and I call that difference delta. And then for each lake and each time point, I ask, okay, well, did the epidemic emerge? And I code emergence as 1 or 0. And then bringing together these two data sets, I ask, does delta predict whether or not an epidemic starts? So here are those delta values on the x-axis. The purple region represents where recovery is higher than exposure, and the red region represents where exposure is higher than the individual's capacity for recovery. And the probabilities from my generalized linear model show a really clear signal that epidemics become more likely as exposure matches or exceeds recovery. And we can go one step further with this and compete this model against models looking at only pathogen exposure or only host recovery. But it's really their interaction, that delta value, that does the best job of explaining epidemics in this system. So neither exposure nor susceptibility alone can predict epidemics. Rather, disease depends critically on the interaction of these two processes. So going back to one of my initial questions today, I don't think it's safe to ignore susceptibility when we try to understand disease in wildlife. So my results show that individual susceptibility and immunity matters for parasites within a lab microcosm, but remains just as important when we transition out into natural ecosystems. Moreover, my results demonstrate that individual variation in immunity can scale up to affect broad patterns of disease, which I think adds more evidence to the idea that intraspecific variation can have really important consequences for ecological interactions. Given the costs and the benefits of parasites, I mentioned that it's important to understand the conditions under which disease spreads. And I think if there's one primary takeaway from my talk today, it's that to understand disease in aquatic ecosystems, we need to first look under the surface and continue looking at how lake ecology influences infection, but also look inside the host themselves, looking at both the parasites as well as the anti-parasite traits that are used to prevent infection. So thank you. That wraps up my research seminar, and I'm happy to take any of your questions. Yes, Patrick.
Super good talk, really enjoyed that.
Thank you.
So you have this whole framework for explaining why epidemics can begin if exposure and susceptibility. Can you also use that to explain why epidemics end? Or is there any?
I haven't gotten to that yet. That's an area that I definitely want to pursue in the future. One thing I can say is I've started to look at why epidemics get as big as they do. And while both exposure and susceptibility are really critical at the beginning, once an epidemic gets going, it's all exposure dominated. And that's because the number of spores that begin attacking them following transmission is so high that there's no normal level of immune defense that can contend with that level of exposure. But as for the end, I think that is a question that Meg and Spencer and Carla and I want to know as well. And I haven't dug into that with my data yet, but very good question. Yes?
How much genetic variation there is within the population of the parasite, and is there a covariance between host and parasite genotypes?
Yeah, so the question is whether there's genetic variation within the parasite. And it turns out that in this parasite, there's actually extremely limited detectable genetic variation. And from talking to ascomycete specialists, I guess that can be kind of common within the ascomycetes. So in this system, that makes it really easy because we can only focus on the hosts themselves and host genotypic diversity. But in other systems, it would be really important to explore diversity within each of the players and any form of matching or mismatching between them. Yeah.
Sorry.
No, that's okay.
So if I understand this right, so hemocytes increase, that increases clearance.
Yes.
Resources increase, that increases hemocytes.
Yes.
But increasing resources does not increase clearance?
In that experiment, it didn't. So infection outcomes were all the same. And part of that might have been because hemocytes and clearance work up to a certain number of spores in the body cavity. But once you exceed that number, there's no number of hemocytes that are going to keep the Daphnia from getting infected. So it could also be a dose thing where I exposed them at such a high dose that they had 10 spores in their body cavity and they just couldn't clear them. But I think in the field, there are signatures that clearance is more effective when resources are high. But that said, what I was talking about that I'm starting to uncover with Meg is that resources might also result in a more penetrable gut barrier. And part of the reason for that is if you want to be able to assimilate and absorb resources, you want a really penetrable gut barrier. And so if increasing resources increases penetrability, they might get a little bit more susceptible, but they'll have better hemocyte responses. So knowing which one is going to win out in natural systems is a really important question moving forward. Yes, George.
Terry, your highest hemocyte response was in December. Is that because they've gone through certain clones or a genotype that has gone through this strong infection? Now you have a more resistant population, so their immune system kicks in right away?
Could be ramped up. So I think that's part of the equation. And then I also think that tended to coincide with turnover that year. And so right as the epidemics end, we saw a lot of great resources coming out, and egg counts went up considerably, and hemocytes went up considerably. So I think a lot of it is energetic. But I have observed some signatures in my data where if you look at the size of an epidemic one year, and then look at the immune responses of Daphnia in June of the next year, there is a relationship where Daphnia appear to be more resistant following a very large epidemic. So great question. All righty.
All right, well, let's thank Tara again.
Thank you very much for coming.